All right, so hello everyone. Thank you for joining today's webinar. We will be starting in just a couple of minutes. And in the meantime, we are waiting. It would be great if the participants can introduce themselves in the chat box. You can let us know about your educational background and your research interest. Right, we are just waiting for the rest of the participants to join. And meanwhile, we are waiting. It would be great if participants can introduce themselves in the chat box, or feel free to unmute yourself and let us know about your education background and your research interest. All right, so without further delay, let's start with today's webinar. So hello everyone, welcome to Mix Logic. Before we get started, let me introduce myself. I am Sri Gauri Krishnakumar, Research Assistant at Biotech. And today we have the introductory webinar for the various summer training programs that we are offering, which cover topics like big data bioinformatics, transcriptomics, biomedical data science using Python, and cheminformatics. And during this webinar, we will introduce you to the Omics Logic Learn portal and guide you on how to navigate it, walk you through our meticulously designed training curriculums for our various programs and the various training levels, as well as the associated resources. So before we begin with today's webinar, for those of you who are joining with us for the very first time, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. And so far, the training programs have been completed by over 24,000 participants from 187 countries in over 300 workshops. And our training coordinators are regularly updating and customizing the curriculum to suit the needs of students and researchers worldwide. And in order to create a clear path of skill development, we employ the Bloom's taxonomy levels of learning in our training programs. It begins with students recalling and memorizing the facts and progresses to understanding concepts and applying knowledge, then moves on to analyzing data and evaluating methods. Ultimately, students reach the highest level of learning wherein they can design and create their own original work. So this is achieved through a combination of online courses, training modules, data analysis tools, curated data sets, and interactive mentor sessions to provide theoretical and practical learning in bioinformatics. To further enrich the training curriculum, we also provide real case studies that demonstrate the practical application of bioinformatic methods and omics data in fields such as medicine, biotechnology, agriculture, and even astrobiology. By using the publicly available data sets, these projects allow students to explore and comprehend various research questions and analysis. The Omics Logic portal also gives students options for analyzing a given data set, that is, code-dependent path and code-independent path. So if you are someone interested in opting the non-code pathway, then you will be given directions to access the T-Bioinfo platform for bioinformatic processing and analysis of data. This platform includes demo pipelines as well as data management and analysis cloud infrastructure to run the bioinformatic pipelines. And different stages of analysis are performed in different sections. We will be covering about the T-Bioinfo platform in just a short while. And after familiarizing themselves with the T-Bioinfo platform for pipeline visualization and analysis practice, students can then apply their theoretical knowledge in real projects. They can assess their understanding through quizzes, assignments, certificates of completion, and tests. And we also adopt a unique training approach, integrating student learning and research. As discussed, initially we focus on building a strong foundation in bioinformatics and omics data analysis. And as students advance, they hone their abilities to comprehend how to carry out literature review, replicate methodologies, and so on. And moreover, students are also encouraged to develop their data exploration skills and formulate unique, meaningful questions based on their research interests. So here are some of the student case examples that I want to share with you very briefly. One of our students, Ruthvi Bachelor, a biomedical science undergraduate student, who worked on assessing the underlying reason for development of gray matter lesions in multiple sclerosis patients. She was interested in publishing and went on to publish her work in Immunogenetics, which is an open access journal that focuses on the genetic research in areas of autoimmune disorders. And another interesting student publication is that of Sipanta De, a genetic engineering student who was interested in studying the effect of malaria vaccine to determine its efficacy and understand the molecular mechanisms underlying the protection that the vaccine provides. He also went on to publish his work in Genomics and Informatics, which is one of the official journal of Korea Genome Organization. And further, you also have the opportunity to participate in poster presentation competitions and win exciting cash prizes. And twice a year, we conduct Omics Research Symposium, which is an international research event that brings together experts from all around the world, including judges. And the student projects are not just limited to graduates and doctorates. For instance, Andre, a high school student at Mexico, presented his neuroscience project at the Regeneron International Science and Engineering Fair, for which he won the Potemkin Prize. And this program can also be integrated as a part of the existing university curriculum. For instance, our Python courseworks were part of systems medicine graduate program at Georgetown University, and the training program ended with students submitting their projects on various topics such as infectious diseases, metabolic disorders, neuroscience, psychiatric disorders, oncology, machine learning, and so on. And these projects can also be showcased for your higher studies and university applications, such as some of our students have done, like Dilara Daikin, who got accepted to the Arismundus IMBRC program in marine biology. Dhruv Mehra also got accepted to a precision medicine graduate program at the University of Manchester, and Anusha TA, who got accepted to a PhD program in metagenomics. So faculties and researchers have also collaborated through our training programs to publish their research work. And our latest collaboration has been with a team of researchers at Noguchi Memorial Institute for Medical Research at the University of Ghana. And they carried out a computational-based study to understand variants associated with sulfadoxin resistance in Plasmodium falciparum. And if you're someone with a passion for science and business, it is also possible to pursue an entrepreneurship and business innovation track, like a team of industrial researchers from Biological Support Services, a biotechnology company in Nigeria did. So they developed a microbiome test kit to improve the fertility of women in Nigeria and improve their chances of carrying their babies to term. And the entrepreneurship track is not only pursued by researchers, but by students as well. So for instance, a group of undergraduate and postgraduate students worked on developing an early detection kit for the identification of Alzheimer's disease. So all of these example projects and student projects are available on the Omics Project Learn portal. So let me walk you through on how you can get started by signing. So here is the link for creating an account. I'm pasting the link in the chat box. So for those of you who have not created an account on the portal yet, I'll wait for a couple of seconds for the participants to sign up. And once you have signed up, I'll be able to show you the various coursework and student projects that are there on the portal. So to create an account, you can click on the create an account button that you see here, and you can enter your name, email ID, and password. And you can also log in using any of your social media handles. That includes Google, Facebook, Apple account, GitHub account, or even Twitter. So with that, I'll just pause for a couple of seconds for the participants to sign up. And once you have signed up, please put a yes in the chat box so that I can walk you through. And if you already have assigned an account on the portal, you can simply put a done in the chat box so that I get a fair idea. All right, Esra has already created an account. What about the rest of the participants? Have you also signed up? Along the way, if you're having any technical issues while signing up, you can mention that in the chat box as well, and our team members will be able to help you. All right, so I'm just going to start by signing in, but along the way, if you want me to repeat any of the steps, please feel free to do so in the chat box, and I'll be able to guide you. So for those of you who already have an account on the portal, simply enter your existing email ID and password and click on sign in. So once you sign in, you'll be able to see the various courses page. you'll be able to see the courses page, where you'll be able to view the various courses and example projects that are there on the portal. So if you're interested to explore, all you can do is go to the courses page, and here you will be able to see a search bar that says looking for a specific course. And if you're someone who's completely new to the field of bioinformatics, then you can try our introductory courses on bioinformatics, such as Introduction to Bioinformatics, Course 1, as well as Course 2, Bytes and More. So we'll be getting to all of that in just a while by one of our team member who will be walking you through the various courses and associated training programs. So here, if you enter any keywords, you'll be able to explore the courses that we offer. And once you create an account, do update your account. And to be able to do that, all you have to do is head over to the blue bar on the top and click on Welcome Back with your email ID. And here you'll be able to see a profile button. So when you click on this, you'll be able to edit the various account features that consist of your name. Make sure that your name is in proper capitalization and that there are no spelling mistakes, because they will be generated on your certificates as well. And you can also add a profile image for yourself and a brief bio that will consist of your educational background and research interest. And finally, you can add your social media handle links. In my case, I've linked my LinkedIn account and my auth ID. And here you can see the various tabs on your profile section that consist of your activity, wherein we can monitor your learning progress. And here under the courses tab, it will currently display the course progress that you're making for the various courses that you have opted. And once you enroll for any of our training programs, you will be able to see the program here. And finally, after completing the courseworks with the required assignments and quizzes, you'll be able to receive the certificates under the Certificates tab. And finally, here on the blue bar on the top, if you click on the Projects tab, it will display the various student projects that I was just talking about in my presentation. So far, if you have any queries, please feel free to post them in the chat, and I'll be able to guide you. And with that, I'll pass on the stage to Sonalika, who will be explaining more about the various training programs that we are currently offering. So over to you.
Thank you so much, Srigari. Can you please confirm if my screen is visible?
Yes, we can see your screen.
Thank you so much. Hello, everyone. I am Sonalika Ray. I'm the Bioinformatics Project Manager at Biotech, and today I will be taking you through all the summer training programs one by one. So this summer, we are hosting 4 training programs that are specifically designed for students, researchers, professors, and clinicians. And I will just take you through each one of these programs, mentioning what the topics are for each one of them and what are the resources that we are going to cover. So to begin with, the goal of the Omics programs, it is to provide training in bioinformatics, enabling independent research guided by mentors and peer examples. Developing a growing community with shared passion for data-driven research and appreciation of citizen science. Goal is to enable students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. During all the summer training programs that we would be just proposing, the participants could expect a wide range of valuable features and resources. Firstly, the program offers online mentor-guided training and research experience, which ensures that participants receive personalized guidance and support throughout the training or the learning journey. Additionally, there are mentor and subject matter expert videos that would be shared on the program page that I'll take you shortly, providing further insights and expertise in the field. And to enhance the practical aspect of the program, live bioinformatics and technical support would be provided for the advanced level trainers, enabling participants to receive real-time assistance with any challenges that they might encounter. You will also gain the access to the T-Bioinfo platform, that is a comprehensive tool that facilitates hands-on practical experience in bioinformatics. I'll also take you through the T-Bioinfo platform shortly in the session and show you how you can make the most out of the pipelines to carry out your analysis. Moreover, the program also integrates open source alternatives that incorporates popular scientific tools and algorithms that are widely used in the field. And to reinforce the learning and assess the progress, there are practice quizzes and graded assignments that are included as well. And upon successful completion of the program, the participants will receive a certification recognizing their achievements and enhancing their professional credentials. These comprehensive offerings ensure a rich and a rewarding experience for participants that would equip them with the necessary skills and the knowledge that is required in the field of bioinformatics and data science. So whether you are a beginner, eager to learn the fundamentals, or an experienced researcher looking to enhance your skills, our training programs caters to all the levels of expertise, and all these three levels comes with different access types that I'll just shortly take you through. Now I'll take you through each one of these programs one by one, and to begin with, the first program is Introduction to Bioinformatics. So for those who come from a non-bioinformatics field, let us quickly understand what is bioinformatics and how do we use it. So bioinformatics is the science of both storing lots of complex biological data and of analyzing it to find new insights, which we use in many different ways. And today's biomedical research isn't about publishing one set of results and hoping other researchers read it. It's about finding patterns in all the forms of data, linking everything that is out there to provide new insights that we can only spot if we can see the bigger picture. So bioinformatics brings us together the data from lots of experiments in one place, so we can ask those big questions and find the answers. And such data from different sources is available. Rather, the availability of such rich sources of data changes the nature of biological research itself, as it is rapidly acquiring the character of data science. So who uses bioinformatics and who is it for? The answer might have been statistician, computer scientists, or engineers. But the truth is that more and more biologists and clinicians are now expected to understand and handle their own data to publish and succeed in basic and translational research. In fact, biology itself is rapidly acquiring the character of a data science. Just mentioned that biology is rapidly acquiring the character of a data science. Billions of data points on genes, proteins, and other molecules is compiled in large files and systematically studied. Analyzing this data leads to more knowledge and understanding about living organisms, which includes human health, biotechnology, crops, and livestock. So this training program will give you an overview of bioinformatics for students, biologists, or clinicians, and the influx of data that is associated with the methodologies and data-driven research that makes bioinformatics for life sciences as prominent as the emergence of computers is for the tech industry. This field is based on high-throughput technologies that generates billions of data points on genes, proteins, and other molecules that characterize biological systems and phenomena. These data, or these different data points, they are compiled into the repositories and can be systematically studied to understand, interrogate, and also establish trends for many applications. Now the question is that is such data available? Yes, and one such repository is maintained by the National Center for Biotechnology Information, which has a collection of peer-reviewed articles, biological data sets, and projects on various topics. So as you can see, in the past several decades, this resource has been growing exponentially. There are over 80,000 publicly available data sets in just one part of NCBI, that is called GEO, which stands for Gene Expression Omnibus. And such data repositories contain data sets on thousands of genes in each sample, with tens or even hundreds of samples per data set. So that's the amount of data that is present. And GEO was just one example. There are more such databases that we need to learn how we could make the most out of them and extract the data and make some meaningful biological insights out of it. So the program that is Introduction to Bioinformatics is designed to highlight the role of big omics data that is shaping the modern biomedical innovation, where molecular data is used for precision diagnostics, big data integration, and machine learning that is transforming personalization of treatment selection and innovative algorithms that allows for unprecedented capabilities with drug discovery or design of large molecules and drug repurposing as well. The program covers some of the important technologies and its application in today's world. So the participants will be able to access these bioinformatics resources along with expert and mentor support to get help with the learning process that will help you learn the transformation of biology to data-driven biology. And as a result, you will learn about important bioinformatics concepts and applications, various applications of next-generation sequencing, role of data science for biologists, and the industry challenges that are addressed by big data informatics. Next up is Transcriptomics for biomedical data analysis. That is an online training program for students, researchers, and clinicians. It addresses one of the major challenges in RNA-seq analysis that is related to accurate, reproducible, and effective processing of large-scale data sets that represents gigabytes or even terabytes of data. And such large quantities prevent many to gain practical hands-on feel for RNA-seq. The training program starts with an introduction to next-generation sequencing and then covers the principles behind it and its applications. The participants will learn about sources for transcriptomic data extraction and gene expression quantification techniques. They would also be introduced to exploratory data analysis techniques and downstream data analysis through differential expression and biological interpretation. And also, they will learn about functional analysis through pathway annotation and a range of analytical tools that could be applied to their own research. And by the end of this beginner level, the participants will have a very good understanding of the key concepts and techniques used in transcriptomic data analysis. Next is the intermediate level that covers the advanced techniques such as pathway annotation, network visualization, as well as regression analysis. You would also learn about machine learning techniques for unsupervised and supervised analysis of RNA-seq data, which includes clustering as well as classification methods. So that was just a very brief overview of Introduction to Bioinformatics and Transcriptomics program. Before I jump into the next programs that includes Python or biomedical data analysis using Python and Cheminformatics, I'll just shortly take you through the program pages of Introduction to Bioinformatics and Transcriptomics and discuss some of the elements of these different levels that I mentioned initially. That was the beginner, intermediate, and the advanced levels, and just look at what resources are really covered in there. So this is the program page for Introduction to Bioinformatics. I'll just share the link in the chat box as well. Those who are interested to get started this summer with an introduction to big data bioinformatics and learn about or get introduced to these different omics fields, this program is the best fit for you. The link is in the chat. You can explore it as well. Now, I am at the interface where if a student has already joined the program, they would see this interface. But if you have not joined the program yet, you won't be able to look at these resources that are up here. So I just wanted to discuss with you these different resources that are put together, and then I'll just show you how to enroll for the program. So as mentioned, in the training program, you have these three different levels: beginner, intermediate, and advanced. You can join any of these three levels based on the type of topics that you really want to learn about, the depth that you want to achieve through this training program, and definitely the time availability. So in the beginner level, we would introduce you to different omics fields. be genomics, metagenomics, or transcriptomics, and single cell transcriptomics as well. And in the intermediate level, we'll dive deep into genomic data analysis. We'll learn about multiple sequence alignment, phylogenetic analysis, variant calling, and also we'll learn more about metagenomic data analysis using the DARA2 pipeline and transcriptomic data analysis from gene expression table to exploratory data analysis to downstream data analysis, which includes differential gene expression and pathway annotation. And in the advanced level, you will learn about these different tools and techniques that could be used to analyze omics data, which includes machine learning using unsupervised and supervised analysis. You will begin with an introduction to data science first, and then you will dive deep into machine learning. You will learn about these different algorithms of unsupervised and supervised analysis. And also you will get a taste of cloud computing and get introduced to programming languages like R and Python that could be used to analyze omics data. So this program, Introduction to Bioinformatics, is a power pack program with an introduction to all the important fields in the field bioinformatics that includes omics, data science, cloud computing, and also R and Python for big data analysis. Now, resources. So on the program page itself, just a second, I just close the different levels first. All right. Now on the program page itself, you can see these different courses that are listed out here. As a part of this training program, you would get the access to 4 different training programs that you can go through and learn about omics and the tools and techniques, and also get introduced to the programming languages. Also, if you click on the very first session, I'll just give you an example and show you the resources that are mentioned. So for each session, the topics have been divided into subtopics, and for each one of them, you have a recorded lecture that you need to go through. You will find the recording of this lecture on the program page itself. It's an embedded video that you can go through. And also you have these links of associated lessons and additional reading materials. Now, what are these associated lessons? They are hyperlinked. If you click on them, you will be redirected to that specific lesson. I just showed you the courses that are present or you have an access to as a part of this training program. These courses have these lessons inside it. And as a part of each session, you are supposed to go through the lessons of this course and complete this course. So if in the beginning we're introducing you to bioinformatics, you need to complete the lessons which introduce you to the field bioinformatics. If it's about genomics, you need to go through this lesson, or transcriptomics or metagenomics, and so on. And you have the lessons present inside each session that you can click on and you'll be redirected to complete this specific lesson. Now, what is present inside a lesson? So inside a lesson, you will find the context, and also you will find images and videos to supplement your knowledge. And you'll also find some additional reading material that could be some educational activities, fun learning activities, and also you will find quizzes and assignments to grade the knowledge that you gain in a specific lesson. Once you complete the lesson, you click on the lesson complete button and proceed ahead. So that was about the associated coursework lesson that you need to complete as a part of the session. And also you'll find the link to the additional reading materials inside the lesson, and also on the program page as well, it's given that you need to go through. Additional reading material has been put together for you, which refers to the research publications and these activities that would be really helpful to you to go through. So we definitely recommend to give a glimpse to these additional reading materials. And if you have time, then definitely go through it properly. All right. Now, in some of these sessions, you'll also find the link to the demo pipelines. I'll take you to one of these demo pipelines in the Transcriptomics program though, but I just wanted to mention here that the conceptual part that you have to take up is present on the Omics Logic Learn portal, but you also have the Bioinfo server where all the hands-on analysis part has to be done. On the server, you have these various sections for transcriptomic data analysis, genomic data analysis, metagenomic data analysis, single cell virology, cancer bio, machine learning, differential expression, GWAS, phylogenetic analysis. A lot of different useful sections are present here. Now here you have these drop-down buttons, which will take you to these different demo pipelines for different analysis. So in the specific session where the mentor is taking you through a pipeline and is telling or teaching how a specific pipeline has to be processed, how to take the data, clean it or prepare it, and then process the pipeline, and how do you interpret the results. For each lecture where the pipeline processing has been shown, you will find a link to the demo pipeline that the mentor has referred to. You are supposed to process this pipeline yourself and interpret the results. And also you will find the link to the data or the link to the data set that has been used in that specific lecture. So you have all the material that is present on the program page, and you can go through it one by one for each and every session. So that was about introduction to bioinformatics. Now, let's quickly go through the Transcriptomics training program page. Before we do that, if we have any questions, you can please put them in the chat box. Feel free to unmute yourself and take up the question. I'll just take a pause here for 2 minutes. If there are any questions, you can please put in the chat box. I would also like to know who all from the participants come from a bioinformatics field or a non-bioinformatics field and are interested to get started with an introduction to bioinformatics. Okay, so Saptarshi Mukherjee, is asking about what is systems biology. So systems biology helps you learn about these different network analysis or pathway analysis. So you will learn about, not in this program specifically, but if you're interested to learn about system biology or learn about how these different systems work through network of genes and pathways and how do they interact, you can enroll in the Transcriptomics program. I'll just take you through that program and tell you where these different concepts are covered and how can we move from gene expression level to understanding the network of genes and how do they interact and which pathways do they affect. Piyush, that's an off-topic question right now, but yes, definitely. I can recommend you about the question that you have just mentioned. Let's just go through these programs and then let's get in touch to have this discussion. For now, if you have any questions related to this training program, you can please put in the chat. And Piyush, also share your email address with us so that our team can reach out to you for the question that you have asked. Shahab is doing his major in microbiology. We have a lot of people here who come from a different field, I'll say a non-bioinformatics field. Saptarshi, so we can take up these technical questions like what is gene therapy or what is systems biology, definitely. We can take you through all of these questions and have you build the concepts in the program. So if there are no specific questions related to program, let's start with the Transcriptomics for biomedical research program here. And I'll just quickly take you through these 3 different training programs. And I'll just tell you what all topics would be covered in the beginner, intermediate, and the advanced levels, and how can you get started, and what are the resources available. Shahab, we are live on YouTube. If you want to refer to the presentation that I just went through, you can definitely refer to the link. Sub-tertiary about publishing, I'll just get back to you on that once I explain the level types. All right. So for the transcriptomics or biomedical research, again, the same thing. You have these three different levels: beginner, intermediate, and advanced. You can take up any one of these levels based on the level of expertise that you want to gain through this program. We'll begin with an introduction to next generation sequencing technology, and we'll discuss how the field bioinformatics really emerged. We'll discuss about how these different data types that we get from these different sequencers, where are they stored? Where are these different repositories through which you can access the data? If you are a student and you are not generating your own data set and you wish to learn how the analysis has to be done, or if you wish to work on a project, there are a lot of data sets that are publicly available that you can download and work on. So we'll discuss about these different sources from where you can download the data sets, and how do you prepare them, how do you clean them, how do you analyze the data and get your biological insights. So once we have a good understanding of different sources from where data could be extracted, next we learn how to quantify gene expression tables from the FASTA files or the FASTView files or the raw data in general, and then how to carry out exploratory data analysis and downstream data analysis, which includes differential expression maybe, and pathway analysis or pathway annotation, network analysis, all of that. This is covered in the beginner level. Now, in the beginner level, we would go through all of these concepts with regards to practical on the T-Bioinfo server, and how do you process these pipelines and get your results to make meaningful biological insights out of it. And also, we will learn about some open source softwares or tools that you can use to analyze transcriptomic or RNA-seq data. Next is intermediate level. Now in the intermediate level, we will go from pathway annotation to visualization of the network of genes and also the pathways. And we'll also learn about regression and factor regression analysis. And we'll take up some case studies to have a good understanding about these different concepts. And also we'll learn about machine learning, unsupervised and supervised analysis for RNA-seq data. So that was about the intermediate level, and advanced level is completely around the programming languages R and Python. So far in the beginner and intermediate level, you will learn about how you could use the automated pipelines for analyzing RNA-seq data, and also how you can use open source tools and softwares to analyze RNA-seq data. But in the advanced level, you will learn about how programming languages R and Python could be used for the data analysis. What are these different libraries and packages? How do you install them? How do you work with them? What are these different types of command lines that could be used? And to mention the number of sessions, in the beginner level, you have 6 sessions, intermediate, you have 6 sessions, and advanced, you have 12 sessions. And to talk about the time duration, it would take you a month to complete the beginner level, a month for the intermediate level, and a month for the advanced level. So that is the time division for all 3 different programs. Now, if you wish to enroll for the program, on the program page itself, I'll share the link of the transcriptomics page in chat too. On the program page itself, you can find these 3 different levels: beginner, intermediate, and advanced. And you can enroll in the program in the level that you would wish to. And if you have enrolled in the beginner level, let's say, and you wish to upgrade your level to intermediate or advanced, you have that option as well. As you can see that right now, I am enrolled in the beginner level for this program. I have the freedom to also upgrade my level to intermediate or advanced. That's possible on the program page itself. Right now, we have some early bird discounts that are up for all these 3 different levels. So if you're interested to enroll in the program, you can email us with the level that you're interested to enroll for, and we'll be able to share with you the coupon code that you can use to avail the discount. So that was about introduction to bioinformatics and transcriptomics. Any questions so far? I can see some questions. I'll take a pause for a minute here and answer the questions first, and then we can learn more about the Python and Cheminformatics program. So Babakar is saying that he's a student in plant biology and breeding and needs to understand gene expression analysis through bioinformatics tools. Yes, then I think transcriptomics is the perfect program for you. And in the beginner level, we are explaining, I just went through the topics. In the beginner level, we have from the third session, I believe, yes, how gene expression tables are quantified and how the analysis could be further done. That's been given in the next sessions. Go to this link, Babakar, I'll recommend, and open the different levels that you have and go through these topics so that you have a good sense of what are we covering here, and then you can get started. And also to mention, these programs don't have a specific date of commencement. You can enroll anytime you wish to and get started. These are the summer training programs that have started. You can enroll anytime and get started. Saptarshi, I did not get your point. Ensemble, okay. Piyush, can you please name some of the open source software used? Okay, so when you talk about databases, there are various databases that we have covered like GEO, TCGA, Firebrowse, GDC portal, how to extract data from these open source the databases. And if you talk about the analysis, then not just the Bioinfo platform, though you get the pathway enrichment analysis results as well there with differentially expressed gene list, but we also refer to softwares like Reactome is there, or Ensemble, or Enricher, or David, or X2K Web, or Gene Mania as well for network analysis and pathway analysis. So we'll take you through these tools and also these softwares that you could use for analysis or biological interpretation of the differentially expressed genes that you get. And for drug repurposing from RNA-seq, we will take you through Drug Gene Budget is there. And then you have the iLinks that you can refer to, or the L1000 is there. So there are various open source softwares that will take you through for RNA-seq data analysis. Saptarshi, to understand these different concepts that you have right now as questions of curiosity, you can definitely take up the program. It's not a one-liner answer actually that I can answer you in just one line about the specific question that you are answering. Otherwise, I could have definitely. And to mention what is transcriptomics is the study of transcripts, but that's a very simple definition. If you want to learn more how the specific RNA-seq data is extracted, how it could be analyzed, I'll recommend you enroll for the program and get started. That would be the best way to understand transcriptomic data analysis. Suffer for advanced level, do we need prior? No, Suffer. No, Sahar, sorry. No, Sahar, you don't need any prior Python or R knowledge for the transcriptomic data analysis training program advanced level. I'll just mention why. So on the program page, you have in the program course and project library, the courses that we would cover in this specific program, Transcriptomics for Biomedical Research. Now, if you click on these courses, you would get redirected to the coursework, which includes these lessons that you need to go through. Now, for R and Python, it's not that we jump to the advanced level of RNA-seq data analysis. No, we will take you from the very basics. We'll take you from the very basics of how to install Python, let's say, or how to install RStudio as well. And then how to import data, the very basic functions in R and Python, we'll take you through them first. And we'll also go through the basics or the fundamentals of the computing languages first. And then we'll proceed towards exploratory data analysis and then data pre-processing and statistical analysis or differential expression, all the type of analysis specific to RNA-seq. So even if you are a beginner, you can get started with the program. Okay, Now, let's have a look at the resources of the Transcriptomics training program. While it loads, you can put your questions in the chat. All right. So as I earlier mentioned that I will be taking you through a demo pipeline on the Transcriptomics training program page. Let us see what a demo pipeline is and how you could process data on the Bioinfo server and what type of results you get that you could interpret. So on the program page for the specific program under each session, along with the demo pipeline link and the data set link, you'll also find the link to the databases and the softwares that are used. So as I had a question, I think it was asked by Piyush that what type of open source softwares you would use. You'll find the link of each and every database or software that has been used in the session details itself so that you can open it or install it and go through it. That is covered in the session. Now let's go through one of these demo pipelines and see what does it include. So if you click on the demo pipeline, you would get redirected to the Bioinfo server, the demo pipeline that you can process and see what type of results do you get. This is quite a big pipeline to take up as an example for the session. So I'll just take up the demo pipeline on differential expression. It's pretty short and simple. My idea here is not to really take up the explanation of each and every tool or algorithm that is being used here, but to show how simple and easy it is to process a pipeline on the server. You just need to upload the cleaned data and process the pipeline by clicking on the specific button that has an algorithm or a tool at its backend. And because it's a demo pipeline, you already have the data uploaded at the backend. But when you process a pipeline, you need to select the specific tool or the algorithm and set some parameters. Now, what tools to use, what parameters to use, what parameters to set is what you would be taught in the training program. And then the moment you end the pipeline, as it's a demo pipeline, you'll get the results then and there. But if you have your own data and you process a pipeline, it would take few minutes to few hours, depending on the size of the data and the number of samples. Let us see what do we have in the results that we can look at. Now you will get some results in the downloads that you can download. These files you have in the TXT and the PDF files that we will teach you how to interpret the results from these different files that you get. What do they mean? What type of results do they include and how do you interpret the results from them? And then you have these visualization buttons out here that you like when you click on it. Let us see what do we have here. So we have a heat map that we can go through and we have a volcano plot and a dashboard as well. I want to take you through the plots and the dashboard to show what type of results do we get here. I think I'm facing some issues with my internet right now that I'm not able to get along. Meanwhile, let me take you through the Python training program. If there are any questions in between for the Transcriptomics program, please feel free to put in the chat. Okay, so now let's discuss about the Python program, biomedical data science using Python. So the demand for skilled data analysts and scientists who can derive meaningful insights from the biomedical data has never been greater. And that's where our online mentor-guided training program on biomedical data science using Python comes into play. So in today's rapidly evolving world of biomedical research, the generation of big data has become the norm. And every day, vast amounts of valuable biomedical data is being produced. That holds the potential to revolutionize our understanding of diseases and also pave the way for groundbreaking medical discoveries. However, the sheer volume and complexity of this data presents a significant challenge, and that's where data analysis comes in. So this comprehensive training program is designed to equip you with the knowledge and the skills that you need to excel in the field of biomedical data science. And through a combination of practical assignments, live workshops, and online resources, you'll embark on a transformative learning journey that will empower you to unlock the potential of high throughput biomedical data. So in the beginner level of the program, when we get started with the program, we will first introduce you to the different types of omics data that is present out there. And also we'll start with the basics or the fundamentals of Python programming and its applications to biomedical data analysis. You'll start by understanding the basics such as data types, variables, control structures, functions, and modules. And then you will dive into data processing and cleaning using powerful libraries like NumPy and Pandas. And once you're armed with these skills, you will move on to exploratory data analysis, using visualization libraries like Matplotlib and Seaborn to uncover the patterns and the relationships that are hidden within your data. Additionally, you will also gain an understanding of dimensionality reduction techniques and machine learning using libraries like scikit-learn. And as you progress to the intermediate level, you'll deepen your understanding of clustering and also advanced machine learning techniques that are specifically tailored for biomedical data analysis. You will explore clustering techniques like k-means, hierarchical clusterings, and spectral clustering using Python clustering packages like scikit and scipy, and also learn model validation techniques and evaluation metrics for supervised machine learning models, and delve into advanced machine learning technologies like decision tree, random forest, SVM, LDA, and UMAP, and apply them to real-world biomedical data sets. So that was a very brief overview of the biomedical data science using Python training program. I'll shortly take you through the program page and show you what resources are covered for each and every session of the program. The last program that we have up for the summer is Cheminformatics for biomedical drug discovery. And this program has been put together in the quest of groundbreaking biomedical drug discoveries, and as this field has emerged as a powerful catalyst for innovation. So over these years, the evolution of technologies in cheminformatics has revolutionized the drug discovery landscape. And by harnessing the power of computational tools and data analysis, researchers can now navigate complex chemical and protein databases with unprecedented speed and accuracy. And this training program is designed to address these challenges that are associated with the understanding, modeling, screening, and then applying cheminformatics strategies to improve drug discovery results. Throughout the training program, you will gain practical skills that form the foundation of cheminformatics. So from exploring chemical and protein databases to predicting protein structures, you will develop a solid understanding of the tools and the methods as well that are used in drug discovery. Once you have gained this knowledge, next you would also learn or gain insights into the chemical structures and their representation. You will learn about the basics of drug design and optimization, and learn how to explore the exciting intersection of machine learning algorithms and cheminformatics data analysis as well. And as you progress further, your knowledge will expand, building upon a strong foundation in molecular biology, bioinformatics, and cheminformatics. And here you will explore the latest advancements in the field, such as AlphaFold and virtual screening for hit identification. Now, through these 12 immersive sessions that you will cover until the intermediate level, you would learn about virtual chemical screening techniques, which includes both ligand and structure-based approaches. And you'll also gain a hands-on experience with softwares like Argus Lab, and different protein databases. Moreover, you will also discover the transformative potential of AI in molecular drug discovery and its applications in healthcare. And I got a question in the chat about the publication or a research project. So in the advanced level of this training program, you will stand a chance to propose a research question to your mentor that you could work upon, and ultimately work on a research project with your mentor and have a publication out of it. So that was a brief overview of the program Biomedical Data Science using Python and Cheminformatics. Now I would like to take you through the program page of Biomedical Data Science using Python. Let me just close up the pages that we have covered. All right. So I'll just share the link in the chat. Yes, Saptarshi, you can definitely work on NGS data analysis, and that is what is being covered in the transcriptomic data analysis training program. And in Introduction to Bioinformatics program as well, you be introduced to the field. But if you want to gain an expertise, you can enroll in the transcriptomics training program and learn how you can do that. So I've shared the link in the chat for the Python training program. Now let us look at the components of the program. So you have these three different levels: beginner, intermediate, and advanced. And I mentioned what all topics you would be going through. So in the beginner level, you would be introduced. Yes, goodness, we do have scholarships available for the programs. I'll just take you through it at the registration step. So at the beginner level, we will introduce you to the omics data analysis, different fields of omics, genomics, genomic data, transcriptomic data, metagenomic data, what it looks like. So that you have a sense of the type of data that you have to deal with when you go through each one of these sessions where you are analyzing the data and interpreting biological insights out of it. You'll get started with the basics or the fundamentals of Python, and then you will learn how to explore data, carry out basic statistics, and then visualize data using different techniques, and you would be introduced to machine learning. In the intermediate level, you'll dive deep into all of these different machine learning libraries and packages that you can work with for building a predictive model. And at the advanced level, we'll take you through some advanced topics on feature selection, generalized linear regression model, and also you will learn about network analysis and deep learning. And we would take up some case studies here for different types of data towards the end, where we learn how transcriptomic data could be worked with using Python, or single cell data analysis, or genomic and metagenomic studies. And towards the end, you have a capstone project that you would be working on. Now, if you noticed in the Python training program, you have these collab links that are given for each and every session. So I like to take you through these collabs. So along the session video and the associated lesson and the additional material, you'll also find a collab link that you have to go through and practice it. Meanwhile, the collab loads, I'll just open the coursework, associated coursework as well. So for this program, you're supposed to go through Bioinfo in Python 1 and course 2. In every program, you might have noticed that these two programs are there. The first program introduces you to different omics data types, and the second program, Bites and Molecules, introduces you to different research specialization tracks, which includes oncology, infectious diseases, precision medicine, agriculture, neuroscience, metabolic disorders, so that you know how different omics data could be worked upon or analyzed in different research specialization tracks. So this is for you to develop a basic or a fundamental understanding of omics data and its applications in different research tracks, and then you gain these specialization in different types of courses that you pick as a part of the program. So now what is inside the Python course 2? Inside Python course 2, you'll find these different lessons, and It's not that if you're a beginner, you need to first complete Python course 1 and only then jump to Python course 2. Python course 2 also starts with a basic or a beginner level that you can get started with, and it will slowly take you to an intermediate and an advanced level. You will first learn how to import different types of data, and then carry out exploratory analysis, visualize data using different visualization techniques through box plots, heat maps, scatter plot, and then you will learn how to perform descriptive statistics on it, and then also perform time-and reduction through different methods, and you will learn about different machine learning, supervised and unsupervised, clustering and classification algorithms, deep learning, network analysis, and that is all that you have for the Python coursework. Now, if I open up one of the courseworks, it's pretty interesting to see that inside the lesson, you have the concept explained first. You have the concept explained, and then you have the code block. You have the explanation for each and every code block. Before that, you also have the description of each and every package or library that has been used. And you are supposed to learn about that specific library first, learn about the code block, understand that code block. And once you have a good understanding about the same, then you can practice the same on the Google Colab. So we'll share these Google Colabs. They're there on the program page. You are supposed to create a copy of this Google Colab and practice it along as you go through each and every session. And we'll have the access to your collabs. And if you have any doubt or any error that comes up in your collab, you can share the link with us and we can help you rectify your error. That is why we recommend working on collab so that we can help you rectify your errors. So that was about the resources for the Python training program. Now let us see what do we have for the Cheminformatics training program. I'll share the link in the chat box. If there is any question, you can please put in the chat. Before I go to the Cheminformatics program, I would like to answer Goodness' question on the scholarship that is available for the programs. Yes, we do have scholarships available for the program. For all three different levels that I've explained, beginner, intermediate, and advanced, you have different scholarships available. You can email us on support@omixlogic.com and we can help you with the specific type of scholarship that you can take up. You need to mention what type of level you want to enroll for, and then we can help you with the code. All right. Now for Cheminformatics training program, you have the levels here and you have the specific types of sessions and the topics that we would be covering here. Now, what different you will find here along the session videos and associated lesson and additional material is the link to the data file. So Cheminformatics for Biomedical Drug Discovery program includes these hands-on sessions. Every session is almost a hands-on session. In the beginning of the session, you have some concepts explained first, and then you have a hands-on practical towards the other half of the session. So you have the link of the data file that the mentor has referred to in the session lecture. And also you have the link to the database or tool or software that has been used here so that you can download it or go through it and then also complete your practical hands-on alongside. And you can go through these different types of topics that are covered at the beginner, intermediate, and advanced steps. I won't be going into too much depth there as we are short of time right now. And I just take you through the resources that we will be covering here. We have the structural biology and cheminformatics coursework as a part of this training program that you need to cover. And for structural bio and cheminformatics, we have covered almost a lot of tools and softwares that could be used from target structure, prediction to docking. We have covered the explanation and how to explore these protein structures through different chemical and protein databases. And we have covered each and every topic from the very basics to an advanced level. So even if you don't have any background in structural bio or chem informatics, you can get started with the program. And in the program, we also have a few lessons. You can see that there is one lesson in R and one lesson in Python and 2 lessons in R. So you can go through the basics first if you are a novice, and then you can go through these Python and R code blocks to understand how AlphaFold Python. So that was about Cheminformatics and Structural Biology coursework. Are there any questions? You can please put them in the chat box. Now the question is, how do you enroll for the program? So if you wish to enroll for the program, we have shared the link in the chat box. You'll also find the links of the training programs in your email address. If you scroll down on the program page, you'll find these 3 different levels: beginner, intermediate, and advanced. You can reach out to us for the scholarship code or the coupon code, and we'll share it with you. Once you have applied these codes here, you will get a scholarship to enroll for the program. And you simply need to click on the Buy Now button, and that will take you to the checkout page through which you can enroll. If you face any problem or any technical issue with the payment gateways, you can reach out to Mr. Ojasri Datta on dev@pine.bio, and he will be able to assist you with the same. So that was how you can enroll for the program. Saptarshi, please share the specific program that you wish to enroll in. Which level do you want to enroll for? And our team will share with you the scholarship details and the coupon codes. You can email Shubham on support@omicslogic.com, and he will assist you with the same. All right, so I'll take a pause now. Are there any questions? If anyone has any questions, feel free to put in the chat, or you can unmute yourself as well. If there are no questions, I think we are good to end today's session. For joining today, and you can reach out to us. Our email address has been shared in the chat, and we would be there to assist you. Thank you. Have a nice day.
Thank you so much, everyone. Have a great day. Bye.